Okay, so now we're going to talk a little bit more about chromosome structure and replication of cells through mitosis. So if you look at the basic chromosome structure, and it starts out here with a double helix. What happens is this DNA double helix gets wrapped around histone proteins. And there are many different types of histone proteins. Some of these are going to allow access to the DNA for things like transcription. Others are going to wind it up tightly and they're not going to allow transcription to take place. So what happens is you'll get these wound up into a nucleosome. This is the DNA and histone complex. They get wound up on top of each other. You're going to get this euchromatin. Euchromatin can then be continued and folded up even further, and this is known as heterochromatin. So the euchromatin is relatively accessible to a lot of the transcription machinery. The heterochromatin is going to be a little bit more tightly coiled, and the transcription machinery simply can't access the DNA at these locations. If you were to zoom in just a little bit, or zoom out just a little bit further, you can see the structure of a classical chromosome. One thing we hadn't yet talked about in 111 is the fact that these arms have a specific designation. The shorter of the arms is known as the P arm, and the larger of those is known as the Q arm. And those are just standard designations that we're going to give to all of our chromosomes. Some of them, it may be a little difficult to tell which is the short arm, which is the long arm, but for most of the chromosomes that we'll look at, it's pretty easy to determine that. Okay, so all of the chromosomes in our body are located in duplicate. That is, we have 23 types of chromosomes, but we have 2 copies of each of these. And you get one of these copies from your mom, one of them from your dad. These are known as homologous chromosomes, those within a pair. Okay? So they're going to code for the same traits, but you're going to get slightly different versions from one parent to the next. So let's look a little bit closer at this, because sometimes people get a little bit confused on this part. So I'm going to bring back what we just talked about. You can see the P arm here is the short arm. Q arm here is the long arm. Okay? So after duplication, you're going to have 2 copies of this. These are known as sister chromatids, and they're going to be connected in the center here. This is the one from your father. This is the one from your mother. So after duplication, it's going to look a little bit like this, like these little X's. Okay? So where you will find this is after duplication. So let's talk a little bit more about the cell cycle and how duplication happens. Okay, so the cell cycle, the majority of the cell is going to spend its time in a phase known as interphase. During the first phase, G1 phase, growth is going to occur here. You're going to get doubling of the organelles. The next phase is known as S phase, and DNA replication occurs here. So you're going to go from having one single chromatid to 2 of these sister chromatids. Okay. The G2 phase, you're going to get continued additional growth, other proteins that the cell is going to need for the actual division to happen. You may see cells that are not actively dividing, and they're going to be held in what's known as a G0 stage. So many cells, such as nerve cells and muscle cells, they're often not actively dividing, and so they are just in this sort of stationary phase. Mitosis is where the cells are actually going to be dividing. Mitosis means division of the nucleus or nuclear material. Cytokinesis means division of the cytoplasm. So let's talk about these phases of mitosis and review, hopefully, for you. The very first phase of mitosis, what's going to happen, it's known as prophase. What's going to happen here is the chromatin, that's that diffuse DNA and proteins, that's going to be condensed down. It's nucleolus, or that dark spot, that's where the rRNA is produced, that will disappear. The nuclear envelope disintegrates. Another term for this is vesiculates. That's the envelope around the nucleus needs to disappear. The spindle begins to assemble. So these are going to be what's used to help pull apart the 2 new cells. Prometaphase is sort of this in-between phase. What's going to happen is kinetochores are going to develop on the centromere, and you're going to physically attach your DNA to those spindle fibers. So let's take a look. Okay, so in a non-dividing cell, you're going to have your DNA is going to be all loose, diffuse. Okay, when you're actively dividing, you need to package that up into these distinct chromosomes. The reason for that, if you think about, say, a ball of yarn, if you have a ball of yarn that's nicely wound up, it's very simple to separate those balls of yarn into two piles. If you have a ball of yarn that's all untangled, and you have, say, 10 balls of yarn that are all untangled, it's very difficult to separate those into two nice piles. So the cell is just simply packaging it up, making it easier to do this division. So during prophase, okay, you're going to see the spindle fibers forming, the centrioles are forming, all the nucleus and so forth is disappearing. Prometaphase, okay, these spindle fibers are going to be attaching to the kinetochore. That is the central area on these chromosomes. Circling them here. Okay. The next phase is metaphase, and what happens here is the spindle fibers are going to tug and pull, and these chromosomes are going to be in alignment at the center of the cell. The next phase is anaphase, and this is going to be where the sister chromatids split, and you're going to get daughter chromosomes. These daughter chromosomes are going to be pulled to the opposite sides of the cell. You may start to see cytokinesis, or division of the cytoplasm, beginning during this phase or during telophase. So telophase is kind of almost like the opposite of prophase. What's going to happen is the spindle's going to disappear. You're going to get the new nuclear envelope returning. Those distinct chromosomes are going to go back to diffuse chromatin again. The nucleolus reappears, and you're kind of going back to normal here. So let's look at these. So again, metaphase, you've got these spindle fibers connected to the kinetochores. They're tugging from both sides, and they're going to be lining up nicely at the center of the cell. During anaphase, they're going to start to pull towards one side of the cell to the other side of the cell, and these sister chromatids are going to be separated. So half is going to this side, half is going to go to that side. And you're going to see here starting of cytokinesis, that is division of the cytoplasm, is going to start to pinch in. And remember, this only happens in the animal cells. The plant cells are going to be a very rigid cell, and they do not have this pinching in. Instead, they're going to be building a new cell wall in between the two cells. As again, telophase here, you're going to have the nuclear envelope reappearing. You're going to have one copy of all of the chromosomes in each of the two different types of cells. Okay, so now I have a short video I'd like to show you that summarizes the steps of mitosis and cytokinesis.
Mitosis is a process of nuclear division by which replicated copies of a cell's DNA are organized into chromosomes. The identical copies of the DNA are then divided equally between two daughter cells. Early in mitosis, during prophase, the nuclear membrane and nucleolus begin to break down. As prophase continues, the DNA condenses to form chromosomes, while the mitotic spindle, made of proteins called microtubules, begins to form and attaches to the kinetochores on each chromosome. During metaphase, the chromosomes are moved to the central plate of the cell by the spindle microtubules. In anaphase, the microtubules attached to the kinetochores of each chromosome shorten, drawing the chromatids of each chromosome to opposite ends of the cell, while the unattached microtubules elongate, stretching the cell. During telophase, the chromatids, now called chromosomes, reach the poles of the cell as the unattached microtubules continue to elongate. The nuclear membranes begin to reform as the nucleoli reappear and the chromosomes relax as the DNA unwinds. Telophase is followed by cytokinesis, or division of the cytoplasm. In animal cells, a cleavage furrow forms at the center on either side of the cell, pinching through until the original cell is divided into two daughter cells. In plant cells, cytokinesis is different due to the presence of a rigid cell wall.
So now I have a couple of questions. I'm going to allow you to pause after each question, attempt to answer it yourself, and then I will give you the answer.